Good afternoon. I'm Jeremy Berg, Director of the National Institute of General Medical Sciences, and I would like to welcome you to our annual DeWitt Stetton Jr. Lecture. Our growing understanding of the complexity of biological systems presents one of the great challenges in modern biomedical research. We're all used to seeing figures with arrows linking genes, proteins, or signaling molecules to describe regulatory networks or biochemical pathways. Today's speaker, systems biologist Peter Sorger, will guide us through approaches to help us try to understand the complex dynamics of living systems. Indeed, these approaches may represent what it means to understand such complex systems. Dr. Sorger uses computational methods along with bench experiments to develop systems-wide models of cell function that still contain information about how individual cell components behave. These models address how it is that cells with the same genes and the same environment can respond differently to biochemical cues or drugs. Dr. Sorger received his undergraduate degree from Harvard and a PhD from Cambridge University, both in biochemistry. He conducted postdoctoral work at the University of California, San Francisco, where his mentors were Andrew Murray and Harold Varmus. Dr. Sorger has been a faculty member at MIT since 1994, and in 2006 also joined the faculty of Harvard Medical School. I refer you to the program for further details about Dr. Sorger's background and affiliations. I'm proud to say that NIGMS has supported Dr. Sorger's research since he started running his own lab in 1994. Before I invite Dr. Sorger to the podium, I'd like to take a moment to tell you about the highly regarded scientist for whom this lecture is named. Dr. Stetton spent most of his career at NIH serving in 2 institutes and the office of the NIH director. He was perhaps best known as NIH Deputy Director for Science, but prior to that time he served as the 3rd Director of NIGMS. In recognition of his many contributions to NIH and the scientific community as a whole, Dr. Stetton has the rare distinction of having 2 NIH entities named after him. This lecture is one of them, and the other is the DeWitt Stetton Jr. Museum of Medical Research, which was dedicated during NIH's centennial in 1987. Dr. Stetton died in 1990. And now on to Dr. Sorger's talk, which is titled Measuring and Modeling Cell-Death Decisions in Single Cells. Dr. Sorger.
Well, thank you very much. It's really a great pleasure to be here at the NIH, and I'd like to thank Jeremy for this introduction, as well as really to start off by acknowledging the enormous amount of help that I've had from NIGMS and NCI, and the many people who've had a big impact on my research, people like Jim Dethridge and Jim Anderson, who have impacted both the early phases of this, and Jerry Lee and Jim were jointly funding this Cell Decision Process Center, which was one of these P50 centers in systems biology that let us start on the science I want to tell you about today. And so the basic biology, I think, is familiar to all of you, and it has to do with this issue of how it is we control the spatial and temporal distribution of life-death decisions. And in this sort of simple and sort of canonical example of the development of a chick limb or how it goes wrong in a human in syndactyly, we're reminded that most of the morphological structures that we have are formed not only by the correct proliferation, but also the correct death of many cells. And in the case, for example, comparing a duck to a chicken, you have the same proliferation of the digits, you just have a completely different death program going on in the interstitial spaces. So we've been particularly interested in problems in apoptosis and proliferation as they apply both to cancer and immunity. And I'm going to tell you a story today that sort of lies right at the intersection between those 2 things. And specifically, what I want to tell you about is the biology of the trail-like molecule, which is a TNF-type, tumor necrosis factor-type molecule that binds as a trimer specifically to receptors in the transmembrane domains of a large variety of different cell types, and in that context is able to trigger a classic caspase-dependent apoptosis cascade. And that results, this binding of the trail to its receptor then results in the activation of initiator caspases, so-called, caspases 8 and 10, C8 in my little picture here. That then triggers the next step in the process and eventually will go on and kill the cell. And the biology of these molecules is really very interesting. They're part of a fairly complex process of immune editing, which I simplify rather grossly in this slide, but to give you the sense that one of their critical functions, and one that's now being exploited therapeutically, is the ability of these molecules to selectively kill tumor cells. And it's thought to reflect the activity of both NK cells and cytotoxic T cells in recognizing tumor-like cells and then engaging through here a juxtakine mechanism, both trail and its closely related fast-type ligand. Now, one of the things that got us interested in this a few years ago is that this class of molecules, the trail-like ligands as well as agonist antibodies, are now in fairly active clinical development by a variety of large and small pharmaceutical companies. And these are, as this little review would imply on another one of these network pictures, being combined with a wide variety of targeted and classic cytotoxic chemotherapies, and all attempting to exploit this property of the trail-like ligand that it will kill tumor cells relative to normal cells. And as with so many of these molecules, and I pull this out of a few recent reviews, what's of course happened is that they've been efficacious in a small subset of tumors in some patients and not in many others. And much of the clinical development of this sort of first-in-class category of sort of immune suppressive agents or immune suppressive agents on tumors is in fact stuck somewhere in clinical development. And the questions that have arisen in this context include this fundamental question of why some cells are sensitive and others not. It does not seem, for example, to be related to the abundance of the receptor on the cell membrane. How in the world might you combine this with other therapeutic agents to increase therapeutic index? And then I think fundamentally, why is it that some tumor cells really do respond and not others in the context of an actual patient? And can we work out how optimally to design those therapeutic modalities? And the thing that in approaching these, and my background really is as a biochemist, and so it's a particular pleasure to be here for the DeWitt-Stettin lecture, we were struck by the fact that these are all really questions of cellular and biochemical mechanism, and yet the development that is being done to try to understand these in a clinical and therapeutic context is, at this point, almost entirely genomic. That is, to sequence the cells which are more or less sensitive, or to look at their transcript profiles. And so the question we were asking ourselves, and which indirectly I want to try to answer today, is how could we take a problem of this type and really develop the kind of quantitative reasoning around the biochemistry of these cell death agents that could eventually be therapeutically applied. And I'll come back at the very end of my talk to a specific way we're going to try to approach that. So I wanted to first tell you a little bit more about the apoptosis of these agents, and as you're going to see, most of the data I'm going to show you today relates to single cells grown in culture, largely tumor cells, but we also look at primary cells. I want to then spend a little time thinking about the underlying sort of quantitative reasoning that we apply to biochemical processes, and this is really the development of a sort of approach to modeling them mathematically, and I'll tell you a little bit about the strengths and limitations. And then we'll talk a little bit about a couple of the things that you observe in those contexts, and can this quantitative reasoning help us think about how it is you actually go from life to death without spending a lot of time in the middle. Thinking about a lot of cell-to-cell variability, and I'll show you an example that's not genetically encoded, and then ultimately thinking about how it is we can begin to make sort of information about biochemical processes like this accretive, even when they involve radically different sorts of biology and different cell types. So sort of to go back first to the little simple picture that I showed you before, here we're looking at the trail molecule binding to its receptor. It then causes the formation of a large intracellular signaling complex made up of a very large number of interesting proteins that I'm not going to tell you about today. That then activates a critical set of initiator caspases, 8 and 10. Those act directly on caspases 3 and 7, and those then directly cleave the cell, so that after the typical cell is exposed to trail, it will die on the order of 3 to 10 hours later. Now, in almost all cell types, so-called type 2 cells, it's critical that the mitochondrion be engaged in this cell death mechanism. So it's really restricted to a subset of immune cells I'll mention later that they can die by the direct receptor pathway. And in most of these other cells, what happens is that the initiator caspases cleave a molecule like BID, which goes on and then leads to the translocation into the mitochondrial outer envelope of a Bax-like protein, which is locally antagonized by BCL2, and that antagonism holds the system in check until BCL2 can exceed Bax, and that then results in a critical process known as mitochondrial outer membrane permeabilization, whereby a series of molecules then normally resident in the mitochondria flow into the cytosol, and that also impinges on caspase 3. Now, when we first started to study this in cell types, and now very recently in the mouse, we thought it would be interesting to take sort of prototypical, relatively sensitive cells and instrument them in a way so that we could actually look at the dynamics of this at multiple points in the pathway. And we wanted to do that on a single cell basis for reasons that I hope become clear as this goes on. So what we had to do was to take the kinds of biosensors that we've all read about from Roger Chen and really do a lot more design on them to get some degree of selectivity in there. And one of the things one discovers about biosensors if you use them in living systems is indeed selectivity is one of the fundamental challenges. So we designed one that would measure upstream in the process and downstream, both effectors and initiators. We also were able to measure that MOMP reaction itself with a protein that could be translated from the mitochondrion into the cytosol. And then we had the opportunity to come in with systems-wide sort of kind of RNAi on a process like this, as well as the therapeutic molecules that are now being designed to treat patients, usually in a tumor setting. I mentioned before TRAIL and its agonist antibodies, as well as a very important class of molecules which directly antagonize BCL2 family members, of which the prototypical example developed by Abbott is ABT 737 and 263. Now, it turns out there's a lot more complexity to this biology than I'm going to be able to get to today, and I will allude to it a little bit and tell you a little bit how we deal with it. And the specific thing I want to mention for those of you who know this is that, in fact, this Bax BCL2 family of proteins is between 25 and 30 family members. Some new members are actually coming along very recently, and those now coordinately regulate this mitochondrial outer membrane permeabilization reaction. And I'm going to refer to members of this family within this canonical set of categories, namely those targets of the initiators that start the process, the pore-forming proteins like Bax, and the antagonists like BCL2. And in today's talk, I'm not going to distinguish among their individual identities, although that, in fact, is important to understand it overall. So one thing we noticed when we went and took these cells with upstream and downstream markers in them and started to image them, and here a cell goes, in this case, this is an HCT116 colorectal cancer cell line. It goes from a healthy blue to a garish yellow when it activates its effector caspases and get ready to die. We noticed that that was, of course, dose responsive over this range, all of which is well above the biologically relevant range, which is probably around 2 nanograms per mil, but that also it was extremely variable from one cell to the next. So if you simply look at the accumulation of cleaved caspase products in single cells, and here you can see one example of a cell dying, going from really alive. If you replated at this point, experiments suggest that these cells are really fully capable of re-entering a proliferative cycle. By this point, 14 minutes later, they're almost certainly dead. And if you then follow the activation of those caspases, what you see is a series of trajectories where I've flagged one as early, middle, and late entirely on an arbitrary basis. And as you'll see later, these are all clonal cell populations. But if you align these trajectories by the time of the mitochondrial outer membrane permeabilization by MOMP, then you see what is really a completely stereotypical trajectory, namely that those caspases are off for a long time, and then bang, they suddenly turn on, and within about 10 minutes, the cell can die. And the duration of this delay, this is at a high dose of MOMP, in typical concentrations we now use, 2 nanograms per mil, even in the absence of ongoing transcription, this delay can be greater than 24 hours. So there's a long waiting process before you decide to die. And so we can sort of characterize that by the following simple figure to say that there's some sort of a delay period that individual cells have, and that seems to vary. There's some sort of time it takes from when caspases are first active until the cell's actually dead. We call that TS or switching. And then there's some overall efficiency of the process. And I won't have time to show you that, in fact, although you would think all cells are alive and dead, we've in fact identified cells in which by RNAi-mediated inhibition or by bortezomib treatment, you can take them halfway, and we call those living dead. And the living dead have the unfortunate property of having scrambled their genome, and those are cells which actually don't take the process to completion. And I'm happy to talk to anyone about that if you're interested later. So if you now take these two parameters, the delay time and the switching time, the first thing you observe is that this delay, in addition to being dose-dependent, and that gives you the dose-dependent behavior, is really very different from one cell type to the next, and it's very sensitive on the other members of these death-domain ligand receptors. In contrast, the time that it takes from the first manifestations of death to the point where you no longer can give rise to a viable cell are almost completely constant. And this is constant across almost all cell types we've examined and constant across dose and ligand type. And in this case, it's on the order of 15 to 18 minutes. And if anything, we think this is an overestimate of that time. The second thing to tell you about this pathway is if you actually look carefully at the dynamics of the upstream process, you see something completely different than what I showed you downstream. In this case, you see the same variability, and I've tagged an early, middle, and late. But if you superimpose those trajectories, in this case, you see the canonical behavior that immediately after ligand has been added to those cells, that they now activate the upstream part of the pathway, and they hold the downstream part entirely in check. And it turns out this is actually a very difficult piece of biochemistry for the cell to arrange, because the immediate target of the upstream caspases are in fact the downstream caspases that will go on and kill you. And so, strangely, a cell, not yet committed to death, is creating tens of thousands of molecules of a potent enzyme fully sufficient to kill it, but it's holding that activity in check. Now, one of the problems of any kind of sort of live cell analysis of this is that you really have to worry that what we had observed here of biosensors of this type is really not typical of the natural substrate. And so one thing within a kind of sort of quantitative framework that I'll tell you about in a moment, and I won't show you the data here, is that we always attempt to fuse as many different measurement methods as possible. And so the live cell imaging here is backed up by standard flow cytometry as well as by conventional biochemistry. And you'll immediately realize that, of course, the average behavior of, say, 1,000 discs imaged in a cell is exactly what you would expect to see in the Western blot. And it's gratifying that you can show in a dose-responsive way that precisely the behavior we see here by imaging is what we see by flow and by blot. And this also allows me to point out one other aspect of the dynamics that really we didn't appreciate until about 3 years ago. And that is that if you take a population of cells and you measure this entirely on a population average by doing a Western blot, you think that it takes the average cell on the order of 4 to 5 hours to activate its caspases and die. But that's not true. It takes 10 minutes. It's just that the cells don't all do it at the same time. So this is one of many examples in which I think the single-cell analysis gives you a fundamentally different picture of the dynamics than you would get by doing a conventional piece of biochemistry across the entire population. So the question we had to now ask ourselves is, how in the world does this kind of what we now call a snap-action variable delay switch, namely, how does this work? Namely, that the cells are able to wait for a very long period while accumulating clearly within them the potential to die because they've activated at least some of their caspases. They haven't committed to death, and then bang, in 10 minutes, after let's say a 24-hour delay, they're going to make this transition. And here's where I want to tell you a little bit about how we're trying to think about dynamical systems of this type, biochemical systems, in a little bit more quantitative detail. And let's ask ourselves first, where would we start for any such analysis? And what we have are all these pictures, and we call these figure 7 pictures because they're always figure 7 when you submit your paper to Cell. Although in this particular example, the goal is to click through and order antibodies. We've got a large investment in the community in putting together interactomes, which are some sort of depiction of biochemistry at low resolution. And then at the same time, we have obviously these marvelous high-resolution structures here of exactly the proteins I'm going to show you in a moment. And the question that many people will ask, can we move directly from knowledge of high-resolution structure through these kind of pathways in the type of dynamics I've showed you today? And the answer is not yet. Now, why is this a hard problem? And I think when I go and talk to my friends like Todd Golub, and I go in his lab and it looks something like this, I'm really reminded of the extraordinary computational infrastructure that we've built around modern genetics and genomics. And if you were to come to my laboratory and look at exactly the same setting, you would see it looks something like this. So we, in fact, largely use reasoning about biochemical systems that's 100 years old. Michaelis-Menten constants, Lineweaver-Burke plot. I mean, these are antiquated ideas for systems that are, in fact, much more complicated. And we recently, at some cost to our sanity, actually wrote a little paper, if anyone's interested in genes and development, where we go in and directly address the comparison between the dynamical systems approach I'm going to show you today and the kind of classic Michaelis-Menten kinetics you may have learned. And what I'll tell you the bottom line is, one is a very special example of the other, and one which increasingly we think has little application to the biology of actual cells. So how would an engineer think about this? And this was dating back now to Jim Anderson. We'll recognize this when we started our little systems biology center at MIT. Well, you know, what engineers have is large, complicated problems. And they have underlying them schematics universally drawn so that, you know, any engineer could come along and figure out which pipe should be attached where. Of course, they know that because they engineered it that way. They didn't have to discover it. And then critically underlying this is some sort of a mathematical representation. And the existence of that mathematical representation is very critical because it allows you to reason across ideas that are simply too complex for human intuition. Now, when we started out on our systems biology center, we imagined we would go across to the chemical engineers, who are numerous at MIT, and we would find from them the exact tools we needed to think about, let's say, trail biology. And it turned out that that wasn't true. They had virtually nothing to share with us. And the last couple of years have actually been a process of trying to work out what sorts of tools we need to build to do this sort of reasoning ourselves. And I want to spend a few minutes telling you about those. And it's worth starting off the way Dick Cheney starts off on any problem, by saying what it is we actually know. And so first we have the known knowns in this example, and this, for what I'm going to tell you today, would be a representation of the canonical biochemistry. So I'm not going to tell you about problems where we really don't know anything about the potential reaction mechanism, and we have to infer that directly either by human intuition or automated methods, and my lab and others work on that as well, but I'm not going to talk about that. So here's a case where we have a plausible reaction topology. We know many, if not all, of the players, but we have a couple of alternatives we want to distinguish. Absolutely critical, of course, is we don't really know the rate constants from any of these reactions, and we're going to have to deal with that relative unknown sort of information because rate constants much matter. There's a lot of speculating in this community about robustness, and rate constants don't matter and the amount doesn't matter. You're supposed to be robust to those features. If so, there would be no genetics. So I think we can dismiss that as being unlikely to be the case because small changes in the concentrations of critical things can make a very big difference. So the basic logic, which many of you may be familiar with, is we do still start with a hand-annotated diagram of some sort. And so let me just imagine now caspase-8, that very first step, the one that we were in fact imaging in those living cells, coming along, reacting with BID, and creating the active cleaved species TBID. And of course what we do is we can break that down into sort of elementary binding and catalytic reactions, and then what we do is we simply write that out as a differential equation. And the differential equation looks relatively unfamiliar to you, but it is precisely, to some of you at least, it is precisely, of course, the language of mass action kinetics. Now, you could ask me, is mass action kinetics relevant in this thing? Should I be using stochastic simulations? Those are all great questions, and after a couple beers, we can talk about them. Now, one of the things that you quickly realize when you approach any piece of biochemistry like this, and this is actually one of the hardest problems we have, is the word models that, and that's what we call figure 7 there, the word model, or the little pictograms that you get out of Reactome, are in fact extraordinarily ambiguous. And so for the piece of biology that I showed you that structure before, so here's BID coming along, activating the pore-containing protein, which is supposed to auto-activate and multimerize. It's locally antagonized by BCL-XL. And this is essentially the picture that you would pull out of Reactome. But you or I, if we actually were to read the papers in any greater detail, would realize that it was much more elaborate and uncertain than that. And so I'm still showing you a picture rather than the mathematics. What we now realize from David Andrews and work in this field is that there are both processes of translocation that are absolutely essential. There are binding reactions and there are catalytic processes here. And there are processes, and I've dotted one here in the blue case, which is BCL-XL recruiting its own cytosolic species to the mitochondrion, that have not been documented in any published paper, but make a lot of structural sense. And so you might want to put that forward as a hypothesis to be tested. So ultimately, you have to arrive at some prior understanding of that. And what we have found is that really there is no graphical annotation that can adequately encapsulate the hypothesis you want to set. And in the end, that's why you end up with a series of mathematical equations, and the models I'm showing you today use about 100 variables and about 100 equations, and this is a little bit of that line. And the neat thing about this, of course, is it's no more than I showed you before. Its very enumeration makes it seem complex, but the idea is fundamentally simple. This mass action mechanism. And in the end of the day, the nice thing is you can now begin to do simulation models. And you can ask, well, did it matter, at least in principle, whether BCL-XL could auto-recruit itself to the membrane? And you'll see that, in fact, it does appear to matter. In this case, you have no weight. In this case, you have a weight, and that could be consequential. And our goal in building these models is to create experimental observables. So our goal is to ask, is there an experiment we could design from the model that we could then test a specific mechanistic hypothesis? Now, it turns out, in the last few years, we've come to learn that this is, in fact, much more of a dead end than I've told you. Because I've said that the picture is inadequate, and I've sort of alluded to the notion without necessarily having justified it, that the underlying mathematics will give us a lot more precision and allow us to simulate and do some sort of analysis. But in the end of the day, what we're left with here is a completely unmanageable artifact. And that is to say, the equations themselves are incredibly error-prone. Even though my lab now consists of largely physical chemists and computational analysts for these problems, they're really unable to tell you when it is true that this set of equations has been correctly cast and when it hasn't. And if we wanted to test those two alternatives on BCL-XL, somebody would have to go in and hand annotate and chop out the yellow bits of this model. So it turns out that the way we're heading with this is actually to begin to bring in a piece of computer science. And the current thinking, and I'll assert this to you and I can't fully defend it, is that in fact the way biochemical knowledge is going to live in the future is certainly not in databases of the literature, although we may always start there. It's not, I'm going to argue, going to live in some sort of picture because that's inadequately precise, but it's also not going to live in mathematical equations either. And in fact, we now believe it's going to live in specialized programs. And we in fact have written one of these specialized programming languages with some other members of the field. It's written in Python. And this is, in fact, the precise executable version of the biology I showed you before. And the nice thing for those of you who do use programming languages on a routine basis will realize that, in fact, it's pretty easy to read. And it's, in fact, a much more manipulable artifact than what you get out of the equations themselves. And moreover, it's what we call rule-based. And what we're doing with a rule-based environment is we are trying to create reusable objects. Because, again, let me take you back to the genomics analogy. We take for granted that if the human genome was sequenced 5 years ago, that we have that accessible to us in a computational form in its entirety for subsequent analysis. It is not true that if a piece of cellular biochemistry was published in Cell last year, that you have that accessible to you as more than your recollection of that paper. So this is what we're going to create, and I'll just leave you with the notion then that where this is heading is in a completely different kind of way of representing biochemical knowledge, and we believe that ultimately that's going to live in programs. And these programs will be executable structures over which we will be able to do formal reasoning. Now, it is absolutely true that they have to be associated with the data. In the absence of the data, they're also equally meaningless. Now, it turns out, and I'm almost at the end of this little sort of technical diversion, and we'll go back to the biology, that there's another half of this problem, of course, to the question of just how do you cast the reaction mechanism in some mathematical form, and that is how do you find all those unknown rate constants and protein concentrations. And the answer is, to some limited extent, you can measure them, but you are always in a world of what we call parameter estimation. And so the way this works, it's known as calibration, is you have some set of data, you run some simulation on a nominal set of parameter values, guessing all the rate constants from the prior knowledge, and then you measure the difference between your guess and a set of parameters, and you do that repetitively, and you minimize this function. So in the end of the day, what you look for is that set of parameter estimates which are the best fit between the data and the model, and you also impose on them some degree of physical plausibility. And what you get out then is a landscape. It turns out these look exactly like energy landscapes, and they can be handled in the same way as energy landscapes. And each of these, this is a sort of a topographic view. This is an attempt to create a 3D view. Each of those little lines that I showed you over there is associated with a different value, a different probability, and the most probable guys live down at the bottom of the basin. And those are the ones that you're going to use for all subsequent analysis, okay? That's the minimization of the objective function. But if you look at this over the model that I'm talking about today, you're going, and remember this is a 78-dimensional structure, so you need a few beers before you can understand it. And if you were actually to look at this, you would see all sorts of sort of different types of topology. So here's an example where finding the minimum looks pretty good. You can actually find one minimum and you know you're there. Here you've got two alternatives that you can't distinguish unless, let's say, you do another experiment. But in very many cases across biochemical problems, you have landscapes like what I'm showing you here. And remember, you are trying to find your way in a biochemical hypothesis using only an altimeter. And if you're at the bottom of a valley that looks like this, where the altitude doesn't change as it moves, it means you really have no idea where you are. So the final part of this puzzle that I want to leave you with is what we are now realizing is that as we reason across biochemical systems, we have to deal with a very large amount of uncertainty. And that uncertainty lies in this business of the parameter values. And the net result of this is that if you give me a set of experiments and you draw your little picture, and I instantiate it the way I've just argued for the next 10 minutes, what I will tell you is some hypotheses coming out of that formalism are high probability, some are low probability, but many of them are fundamentally indistinguishable. And I can tell you that we are at the state of analysis at the moment in the field that there are many hypotheses put forward as alternatives in the highest profile journals in our land and are distinguished as being one is true and one is not, which in fact on any formal analysis are equiprobable. So this kind of reasoning infrastructure, if we can finally put it in place, is actually potentially going to have a big impact because we think we have more confidence in our experimental interpretation than any rigorous analysis of this type would suggest. So just to leave you with this, in the end, for today's talk, we're going to talk entirely about ODE networks. There is this parameter estimation problem. That means we have to deal, it's biochemistry, we have to worry about how tightly do things bind to each other, and that's partially known knowledge. And we have to ask, how do these things interact? And that also is partially known. And fundamentally, we're trying to move towards an environment in which that degree of belief is codified and handled in a formal scheme. I'll say parenthetically that I've only talked to you about one type of mathematical representation of biology, and there's an entire spectrum here. We've written a few papers on this. I'm happy to share with you, in which you go all the way from really no assumption at all, pure regression, right down to the high assumption world that we're using today, and they all have their strengths, and they might be used in some combination. So let's go back to a little sort of Gedanken experiment before we get to the meat of the biology that I want to tell you about. And the first is to think a little bit about this switch that I told you about. So now we're looking at some live cell trajectories. And what we're noticing is in each of these three individually cells chosen from a whole series done at different doses, is here we are accumulating the cleaved effector caspase. And at some point, the cell senses that there is enough of this molecule here, and MOMP occurs, and we have this very dramatic transition in which cytochrome C, which is effectively what's being assayed in these live cell images, as part of a reporter now flows into the cytosol. And the old idea that came from Stan Korsmeyer now over 15 years ago is that what you have is the accumulation of an effector here, which is a cleaved caspase substrate T-bid, and that goes up to some threshold, and when you hit the threshold, bang, you die. And you can, in fact, image those reactions in single cells. For either of the two pore-forming molecules here, back on the left and backs on the right. One is mainly in the mitochondria before the other one has to translocate. And what you see is a lot of sort of dynamics until suddenly these pores form. And here's going to be a pore-forming reaction up here first. Here you can see these pores forming. And this is actually looking at the oligomerization of either back or backs through which the pores form. And if you quantify this in the second channel, you see the following rather interesting thing is that in the course of a dying cell, the number of pores increases rather dramatically. So you can end up with several hundred pores in the, at this point, usually fragmented mitochondrion of a typical epithelial cell when you die. But in fact, the MOMP reaction, the thing that determines whether you will live or die, which activates the caspases, is over within 3 minutes. So when the first detectable pore happens, bang, the whole thing's done. And the rest of it seems, at least at the current state of understanding, to be epiphenomenological. So how might this work? Well, if you look in the textbooks, and in fact you look at Stan's original imagining of this, what it was that you would have BAX, which is going to form the pore, and BCL2, which is going to be a negative regulator. And you're going to get an increasing amount of activated BAX over time. And it's simply going to accumulate. And there's going to be a resident inhibitory molecule, BCL2, and its other family members in the mitochondrion. And when the amount of BAX exceeds the amount of BCL2, then suddenly there's free BAX. Free BAX is able to oligomerize. We know that now in some detail, and you form the pore. And so the idea might be, here's accumulating BAX, here's the amount of BCL2, when it exceeds, then suddenly the pore opens. And in fact, actually, if you do this, And this is one of the sort of fun little exercises you can do when you begin to build these simulations. You realize that this is completely bunkum. There's absolutely no way to make a switch out of a competing negative and positive regulator. And so I've shown you simulations. We know that the binding affinity here is less than 1 nanomolar even in the membrane, and you'll see that, in fact, there's no switch formed at all. And the reason for that is, of course, as you begin to exhaust the amount of negative regulator, it gets slower and slower in terms of the reaction it's able to do because it's at lower and lower effective concentration. So in fact, even these simple ideas we have about how biochemistry works turn out to actually require much more complicated instantiations. And in fact, in this system, we can go through in simulation and rather more weakly in experiments, this is the cutting edge for us, is now to really try to work out what is necessary to make WOMP a reaction that can do nothing for 24 hours and then go to completion in 10 minutes. And the answer is there's a considerable amount of topological uncertainty. It's very important that this occurs in a membrane because you have very rapid diffusive environment in that case, and it's also important that you have oligomerization. So the biochemistry of a lot of these dynamic processes that we're familiar looking at are actually quite different than the textbooks would have you believe. And again, you could say, well, you know, how do you know how they are working? The answer is we don't know until we do the experiment, but we can rigorously test the hypothesis in the paper, and I can tell you it's not correct. In fact, it's not only not correct, it's physically impossible. And then fundamentally, we think that these are worth pursuing. So the specific application of this methodology, which I've drawn out for maybe a little long, that I want to now address is the thing we started with. And that is, what is really the origin of the variability in this reaction? So we thought that if we understood the basis of variability, we'd have a better chance of trying to understand those things that are constant. And the basic observation that you can make, and we've now made this over dozens of cell types, is you can take a set of cells, and these are almost all epithelial cells, and we've done this with 3 primary human cell types, and it looks to be the same. You treat them, in this case, by exposing them to ligand. You recover the survivors. You let them to grow for a little while. They form another colony. You treat them again, and you recover survivors again. And what's remarkable in this process is, first, you have that variability in when cells will die, and we've looked at that already. And secondly, you always have some fraction of survivors. And in this experiment, we set it up to do 80% kill, so 20% survive. And you just do that by adjusting the ligand. And you can actually now, it turns out, in most tumor cell lines, even repeat this experiment up to 6 to 8 cycles, at which point, of course, you'll eventually accumulate frank acquired resistance. These are genetically unstable, so at some point the receptors will go away, or there'll be an upregulation of something else and it will no longer repeat. But it is nonetheless remarkably true that you have this kind of short-term heterogeneity that can be repeated cyclically in many situations, and I'm showing it for trail and FAS here, but we've seen exactly the same thing, for example, in responses to gefitinib, a drug that targets EGF receptor, or ABT737 itself. So Sabrina Spencer in my lab thought it would be sort of interesting to try to work out where in the response of cells to these ligands or to drugs this variability arose, and she had sort of a simple idea that it either came before she started or it happened afterwards, so prior or subsequent, relative to the timing of ligand. And the most common form, of course, of prior variability would be the cells are just different. These are, tumor cells. They could be genetically different, growing in a dish. The field favored the idea that it was the point in the cell cycle, and most of the people who did mathematical modeling thought this was all stochasticity, so-called subsequent or intrinsic noise. And the notion there is that the extent to which you see variability in any reaction scales roughly as 1 over root n, just like Poisson noise. So the smaller the number of reactants, the greater the statistical fluctuation in their activity. And that was sort of their favored hypothesis. So Sabrina did the simple experiment, which we've now carried out numerous times, of taking a cell, a mammalian cell in tissue culture, allowing it to divide into 2 daughters, at some point adding trail, and then determining when the 2 sisters divide. And of course, because these cells arose from a common progenitor, to the first order of approximation, we know that they're genetically identical. And we could then represent the difference in the time of death of the 2 sisters, delta TD here, relative to the lifetime of the cell. And we could ask, well, was there any correlation? So, for example, were sisters completely randomly distributed with respect to each other? Were they absolutely identical? So let's look at that for cells where they were recently born. So if 2 sisters were born from a common mother within 6 hours, and you look at when those two cells die after trail addition, or FATs, or TNF, and in the presence or not of various drugs, what you find is that it's highly correlated. So the two sisters are nearly identical within the accuracy of measurement of this method. So that tells you it's not a stochastic reaction, because stochastic things are not heritable. On the other hand, when we then plotted this correlation coefficient here over time, we had the interesting finding that, in fact, it decayed away. And it decayed with a half-life of around 11 hours, so that after these cells had undergone 2 divisions in the dish, they were no more similar to each other than any randomly selected members of the population. So what you have is a phenomenon here of transient heritability. And we thought that this could, in fact, and this came from some work of Uri Alan at around the same time, have something to do with the rate at which proteins were being synthesized in cells. Because we know over the short time, the rate of protein synthesis in cells is, in fact, highly variable. And it's highly variable because cells transcribe genes in little bursts. And so we thought that what would happen if we came in and we actually fiddled with the rate of protein translation. And so we did this with cyclohexamide, and I'm showing you the 2 extrema. This case, which I haven't told you so far, was in the absence of ongoing protein translation, where we saw that we had high initial similarity that decorrelated with a half-life of 11 hours. In this case, where we actually allow full transcriptional activation, and we believe the critical genes are, in fact, genes activated by trail receptor itself, then we see that the process decorrelates on the order of 1.5 hours. So this is transcription dependent, and it led us to reason that, well, this wasn't genetic difference, these are sisters, wasn't point in the cell cycle. I haven't shown you that data, but it wasn't, and 5-cell imaging so we can tell, and it didn't have to do with where you were in the dish. We also could rule out the idea that it was all stochastics. So we came to the simple notion that it had something to do with the levels of the proteins in these cells. And so you could simply ask yourself the obvious question, which is, how different if you took an initial clonal population of cells and you grew them up, or you took a normal population and you grew them in culture, and you actually assayed them, how different are protein concentrations from one cell to the next? And to this, of course, you can use flow cytometry, but it turns out you actually have to do some fairly sophisticated calibration of the flow cytometer, because you have to ask yourself, if cells, in fact, were absolutely identical, what would that look like in the flow cytometer? And that's something we know is the impulse function of the instrumentation, and we measured that by the on and off diagonal interquartal variation. It turns out for this experiment, one of the very hardest things to do is to convince yourself that there are antibodies that are selective for the target. And we did that over the relevant range of protein concentrations by overexpressing a GFP, I'm showing this for BCL2, and then also measuring it on the flow cytometer and making sure that the correlation coefficient was 1, and making sure if we left out the antibody that it was effectively 0. And I can tell you, because this field is of clinical importance, there are several dozens of antibodies here that are huge sellers for their vendors, and of the 12 we looked at, only 5 actually go away when you knock out their gene. So that was sort of an interesting thing. So there's already a challenge in getting any selectivity. So then you do it, and you get the flow cytometry profile that you might all expect over a range of antibodies, different means, and you can simply use unit variance scaling to normalize that. And what we now realize is, in an isogenic population of cells, what you have is a log normal distribution, exactly what you're all familiar to seeing in the flow cytometer. And there is now a robust theory from prokaryotes as to why it has this distribution. And remember, it's not normal, it's log normal. That means it's long-tailed to the right, and the typical coefficient of variation is 0.3 to 0.5, and we've now measured this for about 45 proteins. And the variation certainly arises because of transcription and translation. And so that simply tells you what you might have assumed to be true sort of at the beginning, which is, even if cells are genetically the same and on average they have 30,000 receptors, why will they all have 30,000? Some are going to have 29,000, some are going to have 27,000, et cetera. The question, of course, is that variability so measured sufficient to explain the variability in phenotype? And so there's where the simulations are extremely helpful. So you can go in and take the measured variability that I showed you today, and you can run the experiment. That's a re-representation of my very first slide, and you can see in simulation that it's essentially the same, except a little less noisy. So what that tells you is that, in fact, the variability that we measure from one cell to the next in an isogenic population is, in fact, sufficient to explain the phenotypic difference. Is it, in fact, the explanation? And that's actually a difficult problem to get at. And the way we've been getting at, and I'll show you only one experiment of this type, is to go in and actually, in a cell which is either knockout or not, to overexpress one of the critical components in a GFP tagged fashion, and then do multicolor imaging. So we can actually look at the rate of the reaction while we overexpress or underexpress individual protein components. And this is showing you in one channel death, I think, in red, and the level, in this case, of BID in green. And if you plot those data, what you can see is something like this. And the unremarkable fact is that the more BID you make, you die faster. We knew that from Stan Korsmeyer. The important fact for the analysis today is, in fact, the variance around this mean, which is the interquartile range. And the similarity between simulation and experiment we can now use to make a probabilistic argument that the variance in these protein levels is, in fact, explanatory for the variance in overall phenotype. And what we've now learned, and I'll summarize and then begin to conclude here, is that there are actually two sorts of things we have to worry about. We have to worry about what is the protein distribution itself. I've told you the default is log normal, and when it isn't, for example, if you're looking at nanog in an iPS cell population, then that's interesting biology. And then you have to ask how sensitive is the network to that variance, and that actually is dramatically different from one situation to the next. And what we've been able to do in simulation is to go through and look at the variance in each of the components of the network. Here's BCL2 that we were just looking at, or BID, and then to work out what that impact is on the phenotypic variance. And I'll just summarize that by telling you in most cells, and remember variability is the same across all these proteins to the first order of approximation, there's a huge contribution at the receptor layer, a modest contribution at the mitochondrial layer, and virtually nothing downstream. So that is to make the point that even though I think it is true, and it is the default physical state of cells, that genetically identical, they have different concentrations of their reactants, it is not true that that is equal phenotypic consequence. And that's something we're just learning how to actually analyze. Now, why in the world might you have this kind of variability going on? And I think it's important to remember that, of course, the death decision is an all or none. And if you want to have a graded response, you're going to have to do that at the population level. You don't have, in this case, a more or less of death. And so what we're reasoning is that the graded response in a cellular population has to do with the fact that otherwise identical cells will or will not die. And so we think there's actually an adaptive component to this. So I want to wrap up with one last piece of data briefly described and a brief thought. And that has to do with the difference of frank genetic difference. So I've talked about cells which are identical, but of course one of the vexing things in these situations is that, in fact, when you go in and try to understand the function of any individual molecule, you have the problem that it looks completely different in different cell types. And the specific piece of biology we were looking at here actually related to whether you could die with the caspase cascade alone or if you needed the mitochondria. And the experiment that we did was to go in and ablate this XIAP protein. And what we showed in a particular cell type, HCT116 cells, is we could switch them from so-called mitochondrial-dependent type 2 to mitochondrial-independent. And I'll just show you that in one simulation. Here's live cell imaging. Here's the population average measurement. You can see that here, if you take the mitochondria out of the equation in HCT116 by overexpressing BCL2, they will not die. But if you knock out XIAP, they will. And that is, in fact, the measure of a type 1 cell. So the basic idea is that normally these cells need the mitochondrion to die. Here's MOMP happening. And what we did is we took out one of the genes, and bang, the whole thing goes this way by type 1 death. And so we tried to publish it, and we were unsuccessful. And the reason was that there's a whole literature saying that it doesn't matter, that there are many cell types in which knocking out XIAP does not have the phenotype that I showed you today. And moreover, there are many other genes that have been implicated in this type 1, type 2 control. And so we thought here was maybe another case where we could use this computational infrastructure to begin to reason over the diversity of these phenomena. And specifically, we said what we will do is we'll go into the NCI 60, and we will measure the range of protein concentrations for the 20 or 30 proteins that I've talked about in this pathway and the many others we haven't looked at. And I will then do a simulation stepping over each sort of gradation in realistic biochemical concentration and ask what the phenotype looks like. And I will do that with a method called directed Lyapunov exponent analysis. So whenever you're stuck, you look for a famous dead Russian mathematician to help you out. And what comes out of this is, in fact, a landscape. And I'm going to show you one thing, and this is my final data slide. And that is that we realized that, in fact, if you plotted the concentration of XIP relative to the concentration of caspase-3, and we did this over 20 different proteins. I'm only showing you one slice. What we found is we could map individual proteins onto this landscape, individual cell types. So SKW 6.4 cells, classically type 1, they don't care how much XIP they have. The cell type I just showed you where we made a deletion specifically in HCT116 cells, in that case it's absolutely critical, and you go from being purely type 2 to type 1, and then now we found that in fact a very large number of cells live right on the middle here. And these are cells, here's a breast cancer cell line, where some go type 1 and some go type 2. And you can recognize that by single cell imaging, and it's all part of that natural variation. And the analogy that I'd like to make here is to the kind of phase diagrams that we see in the physical sciences. So if you say what matters whether water is liquid or frozen, and if you're an oceanographer, it's temperature, and if you're a glaciologist, it's pressure, right? Because the glacier is the same temperature, but a different pressure. And the answer here is people asked us, well, what does it matter? Is XIP important or not? And the answer is, of course, it is, and it's entirely context sensitive. And we'd like to believe that we are at the verge of being able to create a rationalization and at least a predictive capacity down the road for that sort of context sensitivity. So then just wrap up. I talked a little bit about apoptosis in single cells, quite a bit about how we might be able to develop modeling, a little bit about how you build a switch. And I want to conclude by just reminding you of the different sources of variability in this type of reaction. And I've really focused on the proteome, not because we don't think genome and environment aren't incredibly important, but in fact because we think that this is an additional fact. And the question that we really have in mind is, could it be that this intrinsic variability, which we really don't know how to otherwise alter because we believe it has a direct physical basis, is in fact the basis of the ubiquity of fractional kill? And you see that in all chemotherapies, that in fact there are always some survivor cells, and those will typically repopulate, and you'll see that in vitro experiments. Now, eventually you're going to get frank genetic change, but maybe there's actually something to be understood here in terms of immediate therapeutic response. We have three final goals that I want to share with you, and then open to questions. And with what I've shown you today, other than validating and expanding it, the first thing we want to do is really develop this as a means to do better experiments. And I want to show you a quick example of this. I talked a lot about before the uncertainty in parameters. That is, you do an experiment, you make a whole bunch of live cell observations, you don't know what the rate constants are. Well, you can quantify that uncertainty, and you could ask, how could I get less? And the experiment we imagined here is you went to Dharmakon and you ordered a series of, and this is a computational analysis as yet, you ordered a series of RNAis and you selectively went through the 30 proteins in the network and you whacked each one down by 75%, which Dharmakon tells us is easy, and then you measure the uncertainty. And the good news is the way you get around uncertainty is by doing experiments, okay? So with this in mind, we can ask, well, what is the best order of experiments? And there is an optimal order. And it turns out that the first 5 RNAis, in fact, get you all of the information that you would ever get by doing the same set of 28. But no biochemist doing this or biologist doing this is going to just do RNAi. You're going to go ahead, you got dose here. Why not do dose response? So we imagine now simulating what happened if we did high, low dose, and in this system, 2 different ligands. And now what you're going to realize by bringing a heterogeneity of experiments and optimal experiments, you'll realize the first 2 of them get you more than a completely systematic RNAi-based analysis do. And so, although this is still sort of analysis in its infancy, all engineering is based on optimal experimental design. You only measure what's going to be valuable to you. And I'll tell you, from this one observation over one cell type, I'm going to learn that in fact systematic RNAi-type experiments make no sense in this case. There is an optimal order. That optimal order is determined by the hypothesis, and we may ultimately be able to codify it. Second thing we'd really like to do is to take the types of observations I showed you today and actually relate them to something clinical. And I think that's where the cell death pathways seem most promising to us, because they seem to be directly involved in therapeutic response. And in a collaboration with Tony Latai, he has shown that if you actually measure the susceptibility of MOP to, the susceptibility of human tumors to MOP, and these are actually primary clinical isolates, in this case of myeloma, then you can actually show the degree of sensitivity of the mitochondria is really a very good clinical predictor of response over a wide range, a very heterogeneous experiment. But in fact, we have no idea what Tony's doing in his experiment from a biochemical perspective, and our goal now is to ask, can we put a kind of rigorous mechanistic and quantitative framework behind it? If we could so do that, would we in fact now have a predictive capability that you could bring into a clinic and identify those patients who are and are not responsive? And then finally, goal 3 is to really think about how we actually talk about biochemistry. And I've alluded to some of that today. And the way it would work today is if I was wanting to talk to Jeremy Berg, I'd have some literature that I've read, or I read, actually one review, of course. But anyway, I'd have the literature, I'd have some model in my mind, a word model, I have the experiments, and all I could convey to Jeremy was some really very vague idea of what it was I meant. And if you think about it, what happens if you were doing this in genomics? You wouldn't come along, I wouldn't read out the base pair sequences for you. I mean, it would be completely absurd. You take absolutely for granted that there's an entire infrastructure out there. So even if I told you my conclusion, you could go back to your own laboratory and reproduce it. And so the thing we'd like to move towards, and I can tell you we're only in the infancy of this, is really to be, yes, of course we're going to talk about word models, but in fact we'd like to be able to put in place the kind of computational infrastructure that I very vaguely sketched out for you today that could then allow that word model to be supplemented by knowledge of the experiments, of the underlying reasoning, and then you could make your own conclusion. And in this you'll see that Jeremy's concluded the high probability conclusion is completely different from mine, and he's almost certainly in that case correct. So the people who led this effort in my lab were graduate student John Albeck, Sabrina Spencer and Debbie Flusberg, 2 postdocs, Suzanne Godet and John Burke. Suzanne now has her own lab at Dana-Farber. John Burke is the head of systems biology at Boehringer Ingelheim. And Sabrina is on to a postdoc at Stanford, as well as a much larger range of collaborators, including my longtime collaborator, Doug Laufenburger. And all of this, really, that I've shown you today was funded by the Systems Biology Center grant from GM, and now supplemented by some additional grants from NCI. Thank you very much.
Questions for Dr. Swartz?
Hi. Thank you. That was a very wonderful talk. I have 2 quick questions. One is, what cell lines were you using in these cells? And
yes, so it was diverse, and I can send you some of this is published, some isn't. In general, the imaging experiments we did, obviously, you always start in the yeast of mammalian biology, namely HeLa cells. So the specific movies I showed you in HeLa cells, but we've done exactly the same thing now in MCF10A cells, which are a non-transformed breast cancer epithelial line, and there are 3 successively more transformed variants of that cell line. And we've used those, and then we've also done it in HCT116 cells. The other experiments where I sort of plotted, landscapes, those have a much greater diversity of cell types, typically epithelial cells, however.
so the second question is related to this. So I'm doing similar experiments on a different line of argument, but in our case, cells are moving a lot. So in many of your movies, you did not have 100% confluence. Still, cells were not moving. So are you immobilizing them, or are you doing some treatment?
No. So yes, you're absolutely right. So the movies I showed you were HeLa cells, which are fortunately fairly static. Every other cell type we've looked at moves a lot. And in fact, it not only moves a lot, it moves a lot when you expose it to trail. And trail, in fact, has the rather unfortunate property, I think, for a future therapeutic, that it actually induces a dramatic cell motility, and it actually induces epithelial mesenchymal transitions as well. So in that case, it's a much harder imaging problem. So what we do there is we actually track individual cells using point tracking software. And so the cells are moving around. So all those, that's why I didn't show those, because it's actually impossible by eye to pick them out. Very recently, we've decided that actually tracking is not adequate to that task, so we've moved to micro-pattern substrates. And so we actually have micro-pattern substrates where you can have 6 cells on a pad, and they will actually chase each other around the pad. And so they will continuously move around the pad, and because they're it's almost like they're like a little sequence, you can actually keep track of them. So in fact, tracking is a big problem, and I would say it's yet another area, Jim Deathridge might be interested, it's yet another area where we really lack the infrastructure for doing something fairly simple. General purpose, high quality cell tracking tools are really only recently being developed. Okay, if you email me, you can have the code we have.
So I was struck by the fact that you showed us a very complicated mathematical model with, say, 100 variables, and then you said it was useless because it was intractable. And the story you ended up with for your snap action switch was actually something very simple. It was a simple switch with lots of heterogeneity. And in fact, the same idea has come up in other contexts, like to explain dose responses of secretion of hormones. So my question is, did you arrive at this simple model, which is something anyone can understand, by thinking in terms of the 100 variable model, or did you step back from the problem and just think about the global dynamics?
A great question, and I think I'm sorry, I may have misspoken in a few ways. First, let me speak to the issue of the switch. It is, in fact, is not simple, right? You can describe it simply. You can put a heuristic on it, which was a little, I think I may have shown that early in my talk, a little 3-variable heuristic. But if you want to understand that biochemically, we, in fact, have not been able to fully reproduce some of its key characteristics with models having on the order of 50 species and 100 variables, right? So, in fact, I would argue there is a simple piece of phenomenology under which there is a very complex piece of mechanism. And if you own a very expensive stereo, you'll appreciate that that's the case. Making something linear is actually really hard piece of engineering. So, in fact, there is no simple model. And, in fact, the very simple model there, you know, the Korsmeyer competitive inhibitor, I set that up as a straw man and said that it doesn't work at all. What we can do currently, the hard things in the switch is to stay off. It's actually very hard to stay off when you're accumulating a lot of caspases in your cell. That we can achieve. What we can't achieve is actually what we call perfect rectification, which is whether you die early or late, you die at the same mean threshold in the response. And that's actually a very difficult thing to achieve. Generally, when you die late, all the models tell you you want to die at a lower threshold. Okay, and then the second thing when I said that the very complicated model was useless is, again, I think I misspoke. What I was saying is you cannot, it's not an artifact over which you can reason. And so, I struggle with that in my talk, right? I don't show you models, I show you a bunch of pictures and diagrams, right? So the model is not something, not only that you perhaps can't deal with as an artifact, but it turns out the very people in my lab working on it cannot deal with a 200, 300 equation model. And it's just, it's not actually something we can manipulate. And so we've actually had to develop meta languages, what are called abstraction layers, and so routine, again, in engineering to manage it. So in fact, I don't think what I will now publish in my papers 2 years from now will be the equations. We will publish this as programs. They will actually be programs which you can execute on the web. Underlying them is the mathematics, but there's no need for you to look at it. It's the same thing when you go to your computer, you want to type in your Word document, you don't want to see the machine code, right? And in fact, if you saw the machine code, you couldn't make any progress at all. So it's that very tricky issue in this problem, and you've alluded to it precisely, is where are the abstraction layers? Where do we put a more abstract concept in place? And to be honest, we don't know. We're just struggling with that.
Peter, despite the excellent presentation, I'm still having trouble in grasping the origin of this cell-to-cell variability in otherwise oncogenic cells. What is the origin? Where is it coming from? It's not that you draw the picture, genomes and proteomes and environment. Where is this variability coming from?
Okay, so imagine you have a cell and that there's one cell and it's going to have 30,000 receptors on it, right? Easy to imagine. And it gives rise to 2 daughter cells. And those 2 daughter cells, there is no reason, I would say intuitively, why the rate of transcription and translation in those cells should be precisely the same. And it isn't, in fact, precisely the same. And so as a consequence, one cell is going to make a little more receptor, and one cell is going to make a little less. And in the end, one cell is then going to have 30,100, and the other one's going to have 29,000. And so you will diversify on that basis. The hard thing to understand is, well, among the hard things is why that shape? Why the log normal shape? Because a normal distribution would seem similar to us. And by the way, they're radically different. Normal distribution, 2 logs out to the right, you've got 3 logs out to the right, you've only got 0.1% of the cells. Log normal distribution, 3 logs out to the right with a CV of 0.5, you've got around 8% of the cells. So if you're in a cancer situation, you're asking, where are all those damn survivors coming from? The long tail matters to you a lot. Okay, so why that shape? And that really is theoretical work that's come out of work in prokaryotes. And it has shown that, and this is actually known to be true in mammalian cells as well, is when you have a gene that's transcribed at a high rate, it's not a continuously high rate. We now know it's extremely bursty. So let's say in a 24-hour division cycle for a highly expressed gene like a housekeeping gene, it will be expressed in bursts that are on the order of 10 minutes long, and they're separated by periods of 20 to 30 minutes. So it's made it's made like flashes, and the spacing between those flashes is in fact random. And the reason for that is an initiation complex, in fact, is only present at 1 to 2 copies at any single origin. That is a fundamentally stochastic process for physical reasons. So then the current theory suggests that a process like that, which is essentially a series of spikes that are randomly spaced, driving into a relatively high concentration of the eventual species, because these are highly expressed proteins, 30,000 copies, those in fact will naturally give rise to log normal distributions.
So that in fact underlines your idea of fractional response of cancer cells to drugs. Why are there some cells responding, others not responding? Is because of this cell-to-cell variability?
Correct. And I've told you, and we now have a generously funded program that is in part from you to actually look at this much more systematically, to look at this in a much wider range of drugs. And I can tell you anecdotally, if you kill cells with, if anyone here has ever done IC50 curves, you'll see in some cases, you'll see killing curves which are sigmoids, right? You've got a second order reaction, it's a sigmoidal killing curve. You go from 10% kill to 90% kill over 80-fold range. And then you see all those other ones where it takes you go over 4 logs. Well, almost our imaging currently strongly suggests that's a whole series of little sigmoid curves all piled up on top of each other. And that is natural heterogeneity. And you can take, the important thing to realize, you can take the resistant cells in that, regrow them, and they recreate the entire distribution. It does raise the question, what about things like tumor stem cells? And that's an interesting question. I think a lot of what's ascribed to tumor stem cells is in fact these rapid remixing populations. It doesn't exclude the possibility of long-lived ones as well.
Very good.
Last question.
When you showed the sort of that the variability in apoptosis comes at an early point in the cascade, is that because the number of molecules that's involved early is much smaller? I guess I didn't understand if always at the receptor level that's sort of the lowest expressed protein, and then the downstream signaling molecules have a lot more proteins involved.
Yes. Excellent question. And in fact, unfortunately, I sort of blew through something that's quite complicated. The key idea I wanted to leave you with, and then I'll answer your question, is that even if all the molecules are roughly equivalently variable, and now what I like to go in the literature and find when it isn't, nanog, 2 bumps, that's pretty interesting. T-cell receptors, much more variable than random would suggest. Even if they don't all have the same impact on phenotype. In the HeLa cell simulation that I showed you here, and in experiments, we have been able to establish that it is at the receptor level, and that is not due to abundance. Those proteins are not, on average, less abundant than the downstream proteins. They are obviously less than the caspase and its substrates, but not less than SMAC and XIP. It has to do with really the impact of that variability on the reaction, which is in fact connected to parametric sensitivity. But a complication is other cell types set up elsewhere on my little diagram, SKW 6.4 cells, it's a completely different picture. There it looks like variability can be much more important downstream. So you can't take away upstream, downstream lessons from it, but you can take away the lesson that some things have bigger impact than others. Okay.
Are there any molecules that don't have any variability?
I wouldn't know how to find them, but whenever you see something very precise in an engineered system, think that a lot of effort has been put into it, right? I mean, your Wi-Fi on your computer.
But you haven't found one?
We haven't. Is a big, complicated engineered system. I think if something has a very tight variance, that's because there's very tight feedback, some sort of complicated biology over it. We haven't identified one. Thank you.
Let's thank Dr. Sorger again. Please join us for a reception in the atrium, and next week's talk will be given by Shri Grewal from NCI on epigenetic genome control of heterochromatin and RNAi machinery. Oh, it's in? Okay.